I think Colin Stewart said this a couple years ago, where he said that working with the Barth families and working on this disease with the scientists, but really the families and getting to them, was one of the greatest privileges that he's ever had, and it's true. This is one of the greatest joys, getting to know the families and the patients and other scientists as well. So I don't see this as when you say, oh, thank you for working so hard, I love it. So anyway, so thank you for really bringing joy to my life. So anyway, with that said, I think my positioning in speaking today is probably pretty good. So if you were at the sessions this morning, there's some really elegant, wonderful talks about looking at the pathophysiology in the mouse model, in different disease models, in the inducible cardiomyocytes that we've been really using a lot. So hopefully this is a bridge, this talk, to now what we see in the mouse model, what we see in these iPS cells. Do we see these in the human beings as well? Does the phenotype kind of recapitulated in the human beings? And then this afternoon, you're going to be actually seeing some preliminary data on some therapies, which is really, really exciting, which is really the first time that we've done this since I've been coming here. So I've been working with this group for about 10 years now. Okay. So probably, what's that?
Is it on?
Oh, that would help. Is it on?
There you go.
Okay. So I'm not going to go through the whole background as far as the pathophysiology of Barth syndrome, but really we're interested in what are the clinical manifestations pathophysiologically that we see as far as substrate metabolism and organ function. That's what we're really interested in. So as we know, TAS mutations in Barth syndrome literally leads to these abnormalities of total cardiolipin and elevated immature cardiolipin. And then we know also that because the cardiac muscle and skeletal muscle has high levels of cardiolipin because of the mitochondria, that these are really the tissues that are most affected. Energy production that we saw in a lot of these different model systems using like seahorse and Ouroboros and things like that is significantly impaired. And then this really translates to some of these clinical complaints that we see in the boys, like excessive fatigue, physical activity intolerance, really unable to participate in a lot of different daily activities of life. And really, it's a young disease. We're still learning lots every year about Barth syndrome. So really, how this started, really, Barry Byrne and Carolyn Taylor were the ones who really brought me into this area. And this was the very first study we've done now years ago, and this is Carolyn's article was published a few years ago, looking at exercise intolerance. So really, their complaints of fatigue and exercise intolerance, we were able to really objectively quantify this with an exercise study. And as you see, I'm going to try to here, basically, this is basically exercise tolerance. So this is oxygen consumption, your exercise tolerance, or how you're able to use oxygen to produce energy. And this is work rate, so this is how hard it is to pedal a bike. And as you see, the control, so healthy, unaffected people, they're able to really pedal the bike at a pretty high work rate, and their oxygen consumptions are relatively high and normal level. But you see in the Barth boys, it's really very, very blunted, that their peak VO2 sits at around anywhere between 12 and 15 milliliters per kilogram per minute, and their work rates, really, they only get to about 50 watts, which is pretty low if you go to the gym. And then what really struck me, and that really brought me my interest into metabolism, is looking at this measure called respiratory exchange ratio. All it is a simple measure of expired carbon dioxide over oxygen consumption. And typically, when you're at your peak exercise, when you're working as hard as you can, this level is, this RER, this number sits right around 1.1, as we can see that with the unaffected guys. But it is extremely high in the Barth guys, so very, very high. And what that suggested to me that they're using abnormal substrates, so something that they typically wouldn't, so something like glucose or producing a lot of lactate. So it really made me start thinking about, is it not showing up? Okay. All right. I'm double fisting here. Okay. So it really piqued my interest in looking at substrate metabolism. So this led to me applying to the BSF, which is hugely important, getting this small pilot study, looking at substrate metabolism in Barth syndrome. So what we did is just a small study. We looked at 5 affected boys, really young adults and adolescents, and 5 unaffected guys. And what we did is we basically looked at how they metabolize glucose, how they metabolize fatty acids, and how they metabolize protein or amino acids. And really, so what we found was that it seemed that these guys broke down more protein. So protein was appearing into their blood a lot faster than the unaffected guys. They seem to have a little bit higher glucose disposal rate, so they are able to use glucose a little more effectively than the unaffected guys. But really surprisingly, what we really hypothesized that fatty acid oxidation would be really impaired, but we didn't see that. And why we didn't see that, we think, is because we didn't exercise them or do any kind of physical activity, so it was at rest. So, and then we looked at just really simple associations between protein metabolism and their heart function, see how did these two relate, and we saw a tendency for a relationship. So this really gave us the beginning idea of really looking at this at a much bigger scale in these guys, really quantifying whole body metabolism and how that really relates to heart function. So what we were able to do with the preliminary data from the BSF, so again, very important, because the NIH will not fund you, will give you an R01 without any preliminary data. Sometimes they won't give you an R21 without it either. So that's another story. So we're able to apply and get an R01 to really quantify this in a larger group of people. So this is the title of it. So it's pretty self-explanatory. We're interested in how the heart and skeletal muscle metabolizes nutrients, how that relates to how they produce energy or energetics, and then overall, how does that relate to the function of the organ. So our overall hypothesis was that cardioskeletal, so heart and skeletal fatty acid metabolism is severely impaired, which basically facilitates the need for a different type of a substrate. So cardioskeletal protein catabolism, so basically breakdown of skeletal muscle, potentially heart to get these fuels. Amino acid anapleursis, which basically means just kind of getting a fuel from another source. And then also we thought that they would use more glucose metabolism in order to supply this energy. But because these amino acids and glucose provide really less energy than fatty acids, that we thought that there would be an inherent energy deficit, and this would be related in the energetics, and then ultimately it would result into increased or decreased function of the tissues, especially the heart. So that's what we basically hypothesized, that impaired heart and skeletal energetics mediate this exercise intolerance that we see, fatigue, muscle wasting, the low skeletal muscle mass in these boys, and then ultimately heart failure. So what we did is our specific aims were really to characterize this on a whole body level, to look at heart and skeletal nutrient metabolism in both kids and adults compared to healthy age, BMI, puberty level, and activity level match controls. And then in the adults, we wanted to actually look at heart metabolism as well, and I'll describe how we did that. So not only whole body skeletal muscle metabolism, but also specifically look at how the heart utilizes fat. And we thought that, and we wanted to do, besides only looking at this in rest, which we saw no difference in fatty acid metabolism, we wanted to actually look at this during a low-level exercise bout, because that's what we did. And then we also wanted to, as I said, we wanted to characterize how the heart specifically used glucose, fatty acids, and amino acids as well. And how we did this, we used stable isotope tracers and also radioisotope tracers, mass spectrometry, and PET imaging to quantify this. Okay. And then really, we wanted to examine the relationship between the heart and skeletal metabolism, energetics, and function. So how did these all relate? How did energy impairments relate to generation of cardiac energetics or energy metabolism, and then ultimately function? And how we did that was we used magnetic resonance spectroscopy, really, which is just MRI, looking at metabolites in the muscle and the heart. We used echocardiography, which everyone's familiar with, and then we used exercise testing to look at peak VO2. And then, which I'm not going to talk much about today, is we also did, we wanted to actually look at mechanistic molecular pathways as well, specifically these certain metabolic pathways that we're interested in. So what we did is we end up getting whole blood collection to obtain PBMCs, and we're sending them to Barry Byrne and Christy Pasak to induce them in the cardiomyocytes so we can do a lot of high-level molecular quantification, see if we can kind of relate genotype, phenotype, what we see whole body-wise to what we actually see on the molecular level in the cell. And Christy's going to be talking probably a little bit about that at another point once we get those data. So what did we do? So basically, adults and kids came to St. Louis for either a 3 or 4 day study. So there might be some of you in the audience. It was a pretty intensive study, but we flew a parent and one of the affected kid to St. Louis to participate. They stayed in a hotel that's on campus and participated in a 3 or 4 day study. So usually day one, what we did is we did a peak exercise test to determine peak VO2, and then we did 2 separate nutrient metabolism studies using stable isotopes. And during each study, there's basically a 2-hour basal period where they just laid in bed, and then they exercised at a 30-minute period at 50% of their peak VO2, measured on day 1. So basically, say a moderate exercise level, so something you'd maybe be a little bit of breathlessness if you went out for a brisk walk. So that was basically the intensity level that we did. And And then we measured them for about 60 minutes, again, in a resting period afterwards. And then on randomized, on one day we gave a labeled 13C palmitate, which is a fatty acid, and then we gave a glucose infusion. We added actually an alanine tracer as well, and I can just briefly discuss that in the end. And then on day 2, we used a leucine tracer as well. So basically, we had to do these on 2 separate days because when they oxidize or burn the leucine or fatty acid, we collect it in the breath, and if we give them together, we just can't tell which was which. So we have to give them on 2 separate days. And then we did our echocardiography, and then we did this MRS, this magnetic resonance spectroscopy of the skeletal muscle and heart, which I'll describe in a minute. And then on day 3, the adults only underwent a radioisotope infusion, basically looking at the same things in the heart. We're interested in how the heart used palmitate, glucose, and leucine, using PET imaging. The reason the kids didn't do this is because the PET imaging had a little more radioactivity, and the IRBs didn't really like that very much to have kids undergo that. So this is a diagram of what our study day looked like. Again, on one day they received either the fatty acid and glucose together or the leucine, and they rest for 2 hours with multiple blood sampling and breath sampling, exercise at a moderate level for half an hour. During that time, we get collected blood and breath samples, and then they rested for an hour after that. So this is a picture of one of the participants with his consent. So you see this setup here, he's actually exercising. This is a recumbent bicycle. As you see, this crazy looking thing here is a warming box. It just makes blood draws easier and makes the blood a little more arterialized, since we didn't do an arterial stick. As you kind of see up here, this is the isotope infusions, and we're basically drawing blood and getting breath samples and also using the indirect calorimeter to basically make sure they're exercising at 50% of their peak VO2. And then this crazy contraption here is our breath collection device. So basically, they're breathing in room air, and as they breathe out, the expired breath goes in this collection container. These tubes here are evacuated tubes. We can stick it over a needle, which sticks out of here, which you can't see, and then we're able to put the expired breath on a mass spectrometer and basically tell how much of the tracer that was in the fat or the leucine that comes out in the breath, and we put it on mass spectrometer. We can tell a lot about fatty acid oxidation or leucine oxidation. Okay, so some of our results quickly here. What I did is I combined the adults and the children just because the data look basically the same and only have a certain amount of time to present. So these are combined kids. So control subjects, so unaffected, there's 22 and 24 Barth. Average age is right around 15, a little bit younger for the control. Again, you can see the distribution here, 15 kids here, 14 kids here. I just highlighted a couple quick things. Fat-free mass was significantly lower. I think we kind of know that in the Barth group, that their skeletal muscle mass is lower. Fat mass is also a little bit higher. I didn't highlight that. Peak VO2 significantly lower. This is typically what we've seen in Carolyn's paper as well. Peak VO2 is really very low, associated with really end-stage heart failure, people with ejection fractions that are very low. And then look, interestingly, and again, we know this pretty much, but when you look at standardized heart measures from echo, EF, and fractional shortening, they're a little bit lower, but they're fairly normal. I mean, on the low end of normal. And we also did strain as well, which is a little bit more sensitive way to quantify systolic function and that was significantly lower in the Barth guys, but not hugely lower. So looking at substrate metabolism, so this is palmitate rate of appearance. So this is a measure of fat breakdown or lipolytic rate. And really, we didn't see any difference between at baseline or during exercise or during recovery. So again, this is normalized to fat-free mass. So it didn't look like lipolytic rate really was a big difference in this group. So when we looked at palmitate oxidation rate, so again, this is fat metabolism, how they burn and use fat for energy. We didn't see any differences in baseline. Again, we saw that in our preliminary studies. But during exercise, at peak, 50% of their peak VO2, it was significantly blunted in the Barth guys. So again, a real low level of exercise. The Barth guys were not able to metabolize or oxidize fatty acids to generate energy. And then when they rested again afterwards, there was no difference. So when you look at glucose turnover rate, so really this is glucose metabolism on a real general scale. It seems like it's elevated all at rest, during exercise, and during recovery. Okay, it didn't really even bump up that much during exercise, but really, so it seems like the Barth guys utilized, or at least their glucose metabolism was kind of ramped up a little bit during all conditions, rest, exercise, and recovery. So then when we looked at the protein breakdown rate, which we talked about before, again, this is leucine. We can only look at one amino acid at a time. But leucine's, we use leucine because it's one of the more abundant amino acids. It can be oxidized, and it seems to be representative of a lot of the branched chain amino acids. So, but there was no differences really between at baseline exercise recovery and how basically the body broke down amino acids, or at least leucine in this instance. But when we looked at how they utilize leucine for energy, so leucine oxidation, no difference during baseline, but it was significantly blunted during exercise in the Barth group and no difference during recovery. And that makes sense if it kind of agrees with Bill Poo's work. If the defect is more down chain in electron transport chain, somewhere around ATP synthase or something like that, it just makes sense that all oxidation will be actually decreased. So this, in my mind, this makes sense. And then we looked at plasma lactate concentration at 50% of VO2 again during the same conditions. So really no difference, maybe slightly elevated at rest, but nothing significant. But during exercise, it significantly went up. Again, this is low-level exercise. This is like a brisk walk for you and I down the road. And why do we think this is important clinically? Because these are levels associated with, in someone who is healthy, with like almost peak exercise levels, like you are just about, you can't run or go any faster, so it's a really high level. So we think this is probably contributing, or at least a good marker of fatigue in these boys. Okay, so when we looked at the energetics of the heart, so I'm going to briefly just run through the methodology, because I think that a lot of you know about it. If not, I can talk to you a little bit about it or later. But what we did is we looked at mitochondrial function using basically a magnet, and this is specialized looking at a metabolite 31 phosphorus, so basically phosphorus in the muscle. And we did this using this little exercise device here. So it's like a gas pedal device that has resistance on it. And you push down, basically like you're pushing down on the gas pedal repeatedly to fatigue your calf muscle. And then through magnetic resonance spectroscopy, what we did is we looked at levels of PCR, so phosphocreatine and an inorganic phosphate. And just in a real general sense, these are stores in your muscle that are used for immediate energy, but when you knock these down with exercise, the recovery rate is directly proportional to oxidative phosphorylation. So it's really a surrogate, good measure for the muscle's ability to generate energy through oxidative phosphorylation or through oxidative metabolism. So what do we find looking at this? So energetics, this is basically a little bit of modeling. So we are able to calculate how much ATP through oxidative phosphorylation that these participants are able to generate. So looking at the differences between control and Barth, as you see, the Barth syndrome group has a significantly less generation of ATP through oxidative phosphorylation in the skeletal muscle. So this is basically quantifying what we have seen in these in the mouse model and these iPS cell models. And then when we look at the relationship between energetics of the muscle and exercise tolerance, as you would expect, a very strong relationship. As you see, basically, the Barth syndrome group clusters together here versus the controls. But this is everyone looking at everyone together. But a very strong relationship. But we know that your ability to generate energy through oxidative metabolism and your exercise ability is very tightly coupled, and it seems that way as well. Okay. And then when we looked at the relationship between energetics production and palmitate oxidation during exercise, again, a very strong, or a little bit less strong, but certainly a significant relationship between the two. So if the Barth guys are unable to generate energy very well through oxidative metabolism, and that has to do with their inability to use fatty acids for energy. And then when we were looking at cardiac energetics, we look at this in a little bit different way. We actually just look at a ratio between PCR to ATP in the heart. So really, we don't really exercise the heart. We don't give them a dobutamine or anything like that, or exercise them in the scanner. We just basically look at this ratio, which has been really shown to be decrease in heart failure and a very good predictor of future mortality in non-Barth heart failure. So it's been a pretty good marker. And again, what we found here is in Barth syndrome group is significantly lower than their unaffected controls. So really, this is a good marker of energetic reserve in the heart. So not only in their skeletal muscle, they're not able to generate energy very well through oxidative metabolism. It seems it's the same thing in the heart. And then when we look at the relationship between heart energetics and exercise capacity, again, significant relationship between the two. As we saw in Carolyn's paper as well, that it seems like exercise intolerance is a combination of heart and skeletal muscle impairments in Barth syndrome. And then when we look at the different heart energetics and glucose turnover, so we think one of our hypotheses is if they're not able to use fatty acids, they're probably upregulating glucose metabolism to basically compensate for that deficit. And it seems like there is a relationship between the two of how they metabolize glucose and heart energetics. Okay. And then a couple other correlations I thought were interesting. There didn't seem to be any relationship between cardiac energetics and cardiac function. So energetics didn't correlate with the resting heart function. And I think part of that reason is because we measured heart function at rest. We didn't measure it during exercise. And I think that's really, as far as a clinical outcome, I'm not sure that resting EF or shortening fraction is the best clinical outcome. I think exercise is needed, or maybe that and or strain, even though strain wasn't associated with it. This peak RER that we saw when we were doing testing with Carolyn and Barry, it correlates very well with peak lactate concentration, so that makes sense to us that they have high lactate concentration. They also have a high RER. It's probably a lot of bicarbonate buffering going on. And then palmitate oxidation rate was inversely associated with lactate production. Again, just more evidence that we think that glucose metabolism could be compensating for this lack of fatty acid metabolism. Okay. So I'm just going to quickly go through this because the PET imaging studies are not very interesting, or at least the data aren't very interesting. So what we did basically in the adults is we used radioisotope tracers, so a little bit different than these stable isotope tracers. We gave it to them, and using a PET imager, you can tell uptake, and then taking blood samples, you can measure the radioactivity and tell a lot about oxidation of these metabolites. This is just basically one of the images from one of the guys. This is C11 palmitate, and what you can see here is this is the kind of pictures that we get. This is uptake of C11 palmitate into the heart, and then through some different activity curves, we can tell how much the heart is taking up and how much the heart is utilizing for energy. So I can say that the data were very interesting, but so far looking at 7 Barth and 5 controls, we have a few more to analyze and a few more adults to recruit. But we don't see any differences right now in myocardial blood flow. Fatty acid oxidation rate does not seem to be different in the heart. Glucose utilization, I mean, there's a huge standard deviation here, so there's really no difference that we've seen so far. And then leucine turnover in the heart do not seem to be any different during rest. So what we think is happening is, again, just like exercise metabolism, whole body metabolism, we don't see a whole lot of differences during rest except maybe in glucose, but we think we need to, when you put a little stress on these guys, even if it's a walk down the hall, this is where we see the deficits. So we think we're going to need to do that with the heart to see any differences. So what are we thinking so far? We think lower fatty acid and leucine oxidation rates during Barth. Again, this really agrees with a lot of the preclinical work that we're seeing in these different models. We think that the boys with Barth are compensating with whole body glucose turnover, basically using glucose more during all rest, exercise, and recovery. We saw that lactate production is higher during exercise. That really kind of gives us some teeth as far as fatigue, as far as a measure of fatigue. And then we found that heart and skeletal muscle energetics seem to be related with exercise capacity and also whole body metabolism. And right now we're not seeing a whole lot of differences in the heart metabolism, but we think, again, we need to exercise them to see those differences. So what are we thinking so far? Just some thinking. It seems that, again, agreeing with the preclinical work that fatty acid and glucose metabolism seem to be adequate at rest for these boys. They're meeting the needs for the skeletal muscle and heart, but as soon as you upregulate them with a little bit of exercise, this is where we see the deficits. And we think there's compensating at least through glucose metabolism. We're thinking potentially through gluconeogenesis, and we're looking down those rows right now to see if gluconeogenesis is any different. So we also, in glycogen storage, I have a feeling that glycogen storage is much higher in this group as well. So looking ahead, we hope to complete the metabolic study, so we need about 4 or 5 more adults to complete it, and we should be, this is our final year. We're thinking about adding like an exercise PET acetate study, so when we look at peak VO2 whole body-wise, we think we might be able to do that with an exercise with the heart as well, and we think that might be a good clinical outcome in some of the studies that were proposed. Again, as I mentioned already, we're looking at gluconeogenesis. George Schweitzer received the BSF grant to really look at this in the boys and also in the mouse model. We're completing the IPS studies with Chrissy Pasek at UF. We're doing some endurance. Well, we've completed the endurance, but doing some resistance exercise studies. So Adam Patel, my graduate student, is presenting that work at the poster session. So make sure you stop by and check out his poster. And I think these are really important because I think with any of these interventions, a rehab component eventually is going to be important to get these guys back to really functioning at a high level. And then Collins has this bezafibrate study, which we're excited about, which I think hopefully is going to start soon. And obviously, the ultimate goal here is to get gene therapy trial going. That's our next step, hopefully, with Barry and Christy. So with that, I'm probably about done here. I just want to thank just my group here, Dominic, who is here, Kay, who is here, Barry and Christy is here, and I cannot do it without participants. Thank you so much. So.
At least one question. Matt will have to run around.
Presentation. I'm very curious about how the abnormalities, the metabolic abnormalities is only seen upon exertion during exercise. Is there anything known about the effects of blood vessel vasoregulation? Is there anything known about vasoregulation in Barth patients?
It's a great question. So to my knowledge, no one has looked at this, at least in humans, as far as blood flow. We did a near-infrared spectroscopy study with Barry and Carolyn, and we measured total hemoglobin, which is really a blood volume measurement. It's really not a blood flow measurement, and that was no different. But we are suspecting that there's probably some blood flow dysregulation again, but we have not measured it, and that's something that we hope to look at. It's a good question.
One more.
That was really extremely interesting. It's maybe a bit of a stretch to look at the yeast model, but in many of the studies that we've done looking at the cardiolipin deficient yeast cells, almost the only time that we see a difference between the totally cardiolipin deficient cell and the wild type is during some kind of stress. So in our earliest studies, when we looked at respiratory control and ADPO ratios in the mutant, there was virtually no difference under normal conditions, but when stressed with either increased temperature or making the mitochondria do fast respiration, that is when we saw the differences.
Yeah, right. We see that in the mouse a little bit too, especially with the heart, right? And the heart functions pretty normal unless you stress them.
Thank you very much, Todd. I do think it emphasizes two things. One is you have to stress both the heart and the patient. And the second is that phosphocreatine is a much better measure than just static ATP levels. So thank you.